We had a beautiful ceremony, and we're graced with the presence of our Chancellor and Asante Hene, Otumfo Osei-Tutu II. It was a beautiful ceremony this morning. And for those who weren't able to join us to KCCR for the commissioning of the brand new labs, it was another amazing feat by KCCR. Can we give them a round of applause to KCCR and all the partners who made this possible? Well, I'm going to stand on the existing prayer that was shared by our chaplaincy this morning. And I'm going to officially welcome you to the second session of KCCR at 25. I think we should say hip, 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 hip. Well, thank you so much. I'm going to introduce our chairman for the afternoon. He's a full professor in the Department of Pharmaceuticals at the Kwame Nkrumah University of Science and Technology. He's the immediate past head for Quality Assurance and Planning Unit, and also a former vice dean for the Faculty of Pharmacy and Pharmaceutical Sciences, but currently he's the provost for the College of Health Sciences right here at the university. Please help me welcome Professor Christian Ejare, our chairman for the afternoon. A round of applause for him. Well, Prof is going to give us a very quick word, and then we'll start with our stories. This afternoon is all about how we started then and now as KCCR. So a round of applause one more time for Prof Ejare.
Thank you, Anita. I always prefer to be introduced as the chief servant of the college instead of the long positions and the rest. For now, we are serving the college. I'm highly honored and privileged to chair this section. We are going to have 5 presentations and discussion. And I believe those who are having a program with the resource persons and then the topics have been selected in such a way that it will stimulate some of us and also project what the center, that is KCCR, has been doing over the past 25 years. With this, I formally accept to chair this session. Thank you very much.
Well, thank you so much, Prof. Ejare. As I said, this afternoon we are sharing amazing stories of KCCR. Now we're going to start with how it all developed. Please help me welcome Professor Rolf Holtzman, former director. Oh, you keep the applause coming as I go through. Well, he's a former director for the Bernard Nocht Institute of Tropical Medicine, and he's going to help us with how it all developed. Thank you so much, Prof. Welcome.
So, Anita, thank you very much for the kind introduction. Ladies and gentlemen, dear colleagues, unfortunately, at the time when it all developed, it was not the era of smartphones yet. That's why we are missing photographs, actually. I tried to collect whatever I could get, but unfortunately, I would have loved to have more documentation and illustration of what happened at the time. I will start. It all began actually with a very cruel and terrible event, namely the civil war in Liberia. The Bernard Nocht Institute had what they at the time called outpost in an African country, namely in Liberia, and it was a very colonial setting in a way. The institute had some houses and lands on the compound. Yes, thank you. On the compound of a German iron mining company, and they were staying there. The researchers were staying there in this compound, and occasionally went into the field to do studies on tropical diseases, primarily on onchocerciasis at the time. It was at the BNITM was really like half onchocerciasis research. So, and then the institute had to abandon this kind of outpost because of the civil war. And for the younger people like us, I mean, I was younger at the time, it was kind of comfortable that this happened because we could start all new thing, namely to establish a collaboration with a sub-Saharan African university, and not like an outpost of German territory somewhere in an island in tropical regions or so, but do it all different. And then the director at the time, Hans Müller-Eberhard, on the left, it took me along to search for a new site for this collaborative unit. And we have been traveling most of sub-Saharan Africa. It was very funny because he was very picky. It was the only time in my life when I only traveled business class, and he always was looking for a Sheraton hotel. There are not so many around, there were not so many around in sub-Saharan Africa. And so we ended up here in Labadi Beach. It was the best we could get here. In other places, like he loved Uganda because in Kampala there was a Sheraton. So that was kind of influenced his decision after all then. And so we were in Labadi Beach. So then we stayed. And then the first problem was that we heard in Hamburg from the helminthologists, the warm people. They said there's no onco. There's no onco in Ghana. We can't go to Ghana. There's no onco over there. So I said, yeah, I'm sure there must be onco. At the same time, actually, the head of the medical branch of the German Developmental Cooperation kind of agency, GTZ at the time, what is now is GIZ, he said there are no tropical diseases in Ghana anymore at all, Rolf Korte, so there is no reason to go to Ghana with this. So, and then it's like, we had the younger people, we had 5,000 Deutschmark at the time to prove that there's onchocerciasis in Ghana. And here for the first time, Thomas Krupa comes into the play. He's an entomologist, and together with a clinician, they were traveling through the Offin and Pra River region there at Dunkwa, and studied all in the villages, studied the people for onchocerciasis and the black flies for onchocerciasis larvae. And they found there was indeed, so there was onchocerciasis. So that was settled, the problem. And this is one of the three key personalities in the development of KCCR, which is Thomas Krupa, with his later wife, then Jennifer Evans, and the small daughter Catherine, and the dogs, which were kind of a problem for us, because they weren't really so harmless as he said they were. So we always were kind of afraid of leaving the car in front of his house because the dogs were all around. So anyway, he was helpful from day zero, actually, before. It was in the 1990s, in the early 1990s or then when that happened. Okay, so this was settled. There is onco in Ghana, and then we finally, after the director Müller-Eberhard, who was traveling, and then he quit and went back to the United States. So the major opponent of Ghana had them be removed from Hamburg. So it was easier. The helminthologists were happy because there was onco, and the big opponent had left for the United States, so we could settle on Ghana. After long negotiations with Uganda. So the first encounter then in Ghana was in the office of the Minister of Health, which was Honorable Stephen Obimpe at the time. Now, it was interesting because of him, but it was more interesting because he called then his advisor for Germany, as he said. And they come from a side entrance to his office, come a lady in a batik dress with curlers. So as if she would just come from the bathroom or so, I don't know. Unfortunately, I forgot her name, and Thomas, you don't remember it correctly either. So I forgot the name, but I never forget this picture, sitting in the office of the minister, and there comes a lady from Germany, and she said, and that's why she's a very crucial person in the whole development, she said, you guys, you have to go to Kumasi. That's where the Germans are. So then actually that sent us here. And then we came here for the first time. Then the second important person comes into play. Come on. Can you recognize him? This is George Proby. He was Dean of the medical faculty at the time here in Kumasi. And when we arrived, it was still Müller-Eberhard and myself who came here. And then he said he had prepared everything perfectly. We had an audience with the king at the time, when I also got a tie, I forget it in Germany now, it's older than yours, Thomas. Oh, Bernhard's, yeah, you showed it this morning. And he arranged an audience with the king, and he collected all the institute heads or clinical heads there, and he very proudly asked whether we should have the meeting in German or in English. And not knowing that Müller-Eberhard, who spent basically he was born German, but he spent basically all of his career in the United States. So he spoke English much better, or American much better than German. So I said, no, I prefer English, do it in English here. But George was very proud to say, here we are there. Now this has changed, and I wasn't so happy about it because it looked too Germanish, everything, the Germans here and the Germans there. But we grew, I think KCCR grew out of that stage, and I think it's better now as it is. So it's not so dominated by kind of German influences. So that's what was not the original idea, actually. No, I lost this thing here. Could it be? That's fun. No, I think it disappeared inside here. Yeah, this is a surprise. Can you move it also if I tell you? Yeah? There's a hole. It's large enough to take this remote thing, but too small for me to get. Maybe it ends up here. My pocket? No, it's not in my pocket. No. Okay, you move the thing. Huh? No, we can't close the drawer anymore. Okay. Okay, now I continue. Try to figure it out, otherwise you move the slides. Yeah? Now there was a thing after a lot of negotiations and everything. I was sitting with George Proby in Jofel. Maybe you remember the place there? The airport. Oh, here it is, thank you. In Jofel, and we were discussing how to name the new institute. Also, it was far away from being founded. So George suggested actually Asante Hene Institute for Research and so forth. I had discussed always Kumasi Blue. This is because we found out that, well, other way around. Everyone who has worked in biochemistry knows Kumasi Blue. It's usually written like on the right-hand side, quick Kumasi. In fact, this is derived from Kumasi. The blue is the stain of the Kumasi women. The English just changed the writing of it because according to their language, how they would spell anything that they would call Kumasi. But the origin is Kumasi, is the city here. So, and I was kind of fascinated by the fact that I was in the city where there was kind of the original stain that was generally used for proteins in biochemistry. So I was thinking Kumasi and biomedical laboratories, unified endeavors, or anything with an acronym of blue then. But then George meant that sounds like a nightclub, so we gave it up. So after all, we stick to the conventional one of Kumasi Center for Collaborative Research. Okay. So and then in 1998, after the state agreement had been signed in Hamburg by the Minister of Finance, actually, Pepra was his name, I remember well, there was the inauguration ceremony here in Kumasi. And at the time, actually, at the main door of KNUST, there was this board here that the inauguration of the KCCR. Actually, the first meeting or this inauguration took place in SMS between two wings of the School of Medical Sciences. And we, or KCCR, at the time, had one wing of SMS for laboratories. That's where the work started, actually. And that's also, I'm afraid I haven't even found this one. It's also where, at the time, still famous painter Atta Kwame painted the logo on the wall there. Atta Kwame was quite famous. He had expositions in the Museum of Modern Art in New York, for instance. So he was famous worldwide. And he designed the logo, which is still great, I think. Except that you use now a different red, actually. It must more be a orange red, the original one. And Thomas, you blew it that it was then not preserved as when KCCR moved ahead, so it was destroyed then. Even it was probably, I don't know how many thousands worth painting of Atta Kwame, so it's really not good. So this was the inauguration at the time there, and the Minister of Health, Honorable Eunice Bruckmann Amissa, who also was in Hamburg for the signing of the contract, she held a speech there, on many others actually, which I don't want to go through them now. And this we have seen this morning, they showed it several times. It shows the minister, and behind her, the third key person actually in key personality, it was Frank Kwame. And Bernhard also referred to him this morning. He was actually very important and very, very supportive. Like for 10 years, he was then chairman of the advisory board. He was, as Bernhard said already, that he was trained and became professor of engineering in Berlin, and then moved back to his hometown here to help to establish the university, KNUST. And he was very wise in the decisions negotiated in the early days. You know, the little gritty things between Ghanaian staff and Germans and financing and so forth. He was extremely important. Very humble and nice person. Huh? Come. Hey. Yes. This is the compound where KCCR is located right now. There was a long discussion in the middle of the compound of this giant tree that was standing there, whether it should be preserved or not. And actually, I think we had decided to build the buildings around this tree. But when I came the next time, it was gone. So, but anyway, then what it looked like here, come on, come, yeah. What it looked like let's say April 2003, and then later in December 2005, it was actually the way it used to be, then remain for 10 years or nearly 10 years. Okay. And also, I also want to talk a little bit of science, which is not so known anymore to the present people at KCCR, but I think it was a very important thing. You know, my personal interest is in human genetics of infectious diseases, but there was just had been discovered that genes, certain genetic defect or genetic abnormality is the cause of deafness. And then George Brobby, as an ear, nose, and throat disease specialist, he knew that there's a so-called deaf village, Adamarobe in Ghana, where like 20, 30, 40% of the inhabitants have hearing loss. And so we checked them and we identified whatever a mutation in the previously known gene that caused the deafness in this community there. That's not the point. The point was that we then managed to publish this in the New England Journal of Medicine, you know, high-ranking medical journal. Actually, I found out at this occasion that the value of the accompanying letter, because we wrote to them that, you know, there's a major advance in hearing loss research, but you haven't brought anything of it. So your clients, your readers don't know it. So they took this little. Important is that George was the first author there. And then a year later, he became president of the International Society of Ear, Nose, and Throat Disease Doctors. So in the community, it became clear that having those important papers is not only for paper, but it's really important for the reputation in the scientific community, which wasn't so clear to many of our colleagues at the time, I believe. Okay. Coming also a little bit on our own research, working on the genetics of tuberculosis, and praising, so we had like 2,200 or so TB cases, including X-ray and history and everything, and unaffected controls. And I have to really make clear that a number of people were very important in this here. This was Alice, I can see this case, underneath the KCCR logo, Alice Owusu-Daba and Ed Brown, who unfortunately died in between. He was one of the masterminds of medical research here at KNUST. And then also actually the Health Research Unit of the Ministry of Health in Accra, led by John Giapong. I think he's now, is he Vice Chancellor of some university in Ghana? John Giapong and Abanwa and Ivy Osei. So they were very important in this kind of research. I just show you briefly a result, what we did with those many samples here. Come on. Why doesn't it move? Okay, this is a genome-wide scan. We checked the whole genomes, all chromosomes of all those people to look for difference between TB patients and healthy controls. And these are some signals, nothing important. Very funny that although it is generally known that 90% of the population usually are naturally protected against TB, we couldn't find any gene, even when we took together or collected or joined our data with those from our colleagues in Oxford around Adrian Hill. And what we found though, and I think it's interesting, that one mutation in the toll-like receptor, only for those who know these things, provided 80% protection. It's a very rare occurrence. It's only 1% of the population, but it was a proof of principle that genetics could protect against tuberculosis. Okay, then we did the same thing. Okay, this is this mutation, the toll-like receptor 1 gene causing an amino acid exchange in the toll-like receptor. Anyway, last not least, the malaria studies. Come on. Okay. Same way, 2,500 children with severe malaria complications were studied at Komfanoche Teaching Hospital, and like 3,000 children somewhere in the country. And it was really, it was decisive for the success of these studies that the clinicians here, the pediatricians on the intensive care unit at Komfanoche Teaching Hospital did a very outstanding job. That clearly was the best ever sample of complicated malaria ever got in the world, actually. And that was absolutely crucial for the experiment. And there was Chiri, he was kind of the, Chiri Akbenjega, he was the kind of the mentor of the young boys on the ward there, and Jennifer Evans, who actually became then, at the time, became then the wife of Thomas Krupper, so it was all in the family. She was very important in actually getting the work being done. And there's a series of doctors who were on the wards there of Komfanoche Teaching Hospital, including like Daniel Ansong, who is meanwhile now the dean, I think, of the medical faculty here. There were some interesting data. Sickle cell trait, of course, protects and so forth, but I don't want to go into these details here. But I want to show briefly other interesting data we got because the group of sick children was so extraordinarily well characterized. If that would only move. Okay, the severe complications of malaria are severe anemia, coma, cerebral malaria, and acidosis, and they were kind of classified accordingly here. And the problem is there's a strong overlap of these three complications. And since, this is annoying here. Since they were so well characterized, we could kind of discern the various complications. It's not so important here. That's the influence of the sickle cell trait. And you see here, the line on top is when there is no protection. And then anything under the line shows some kind of protection. And you see on the C that is severe anemia with or without cerebral malaria, respiratory distress with or without cerebral malaria, and so forth. Here, they're all alike. Sickle cell trait protects against all of the complications. And now it's hemoglobin C, which only occurs in West Africa. And you see here, when you look carefully, you see the protection only on the left-hand side with the open circles. That means it's severe anemia plus cerebral malaria, then it protects. Respiratory distress plus cerebral malaria, and so forth. So only when cerebral malaria is part of the syndrome, it protects. On the right-hand side, it doesn't. So hemoglobin AC solely protects against cerebral malaria, not against malarial anemia or acidosis. And the last slide, more or less. And this is the thing of alpha thalassemia, which I think is even likewise important, I think. You see there is some varying protection on the left-hand side, but on the right-hand side you see protection, the open circles, can only be seen if severe anemia is part of the syndrome. So the thalassemia only protects against severe anemia. So the conclusion, hypothesis, how this could happen. Can we get this going? Okay, this is from a different, just showing you something that some of the children get severe anemia, get it within a few days. You see in the upper panel there, you see this is a longitudinal study of children over 8 months that we followed over 8 months, having regular, every 3 days having regular malaria study, investigation and so forth. And there, one of the children, which is shown here, disappeared from the study group and was then brought to a hospital, and her hemoglobin had precipitously fallen to below 15% or hemoglobin 5. There's another child here who very slowly ends up in 5. And the real severe anemia syndrome is the precipitous fall of the hemoglobin. And when you look at the hemolytic, like thalassemia and this is the Bernhard Nocht Institute in Hamburg. In winter time, there's a little bit snow. So I'll leave it to you. Now, what was on the previous slide was that when you have a hemolytic anemia, a continuous consumption of red blood cells, which are being lysed in the circulation, then you have a continuous increased red blood cell turnover all the time, because you lose blood cells very faster than normal, so you get an increased hematopoiesis, erythrocyte new production. And if you then have a sudden precipitous fall in hemoglobin, the system is already accelerated to compensate for the fall. So you prevent, if you have hemolytic anemia, you prevent the drop below like 4, 5 hemoglobin or so. That could be a unifying hypothesis how all hemolytic anemias protect against severe malaria, and therefore are balanced in the population for malaria protection, and somewhat disadvantaged due to the anemia they produce. So thank you from Berlin, from Hamburg and the Bernhard Nocht Institute for your attention. Now, I was told that I have the honor to give a gift to the children of George Robbie. So, you take over?
Okay, I'll say something. All right, so before Professor Rolf Horstman goes off the stage, we have something little here that we thought he's the best person to hand over to Professor Dr. George Rekubrobe to recognize his contribution to the whole establishment of KCCR. Unfortunately, he wasn't able to join us, but I think he's well represented here for someone to take what we have here to let him know that we really appreciate his contribution to KCCR. And we thought we couldn't do this, but Professor Rolf Horstman will be the right person to do it. So that is the secret behind this little box that is going to go to Professor Dr. George Rekubrobe. So if there's anyone here to collect it on behalf, I think I'm told the children are here. So if Stephen is here, okay, all right, okay. So we can remove it.
Is that Robert? Yeah. Are you Robert? Are you Robert? Stephen, not Stephen, yeah. I celebrated with your father when you were born, I remember. Okay, this is for George. I know that he's in the US right now. He gave me a call the other day, so I think he's doing fine. And
he'd like to take the picture over there, so.
Give it to you. So you have to take it, and I congratulate you, and all the best to George. Huh? The best regards. Okay.
Will you say something, or you want to say something? Okay.
Hello, good afternoon. My name is Stephen Rekubrobe, and I'm the last son of Professor Dr. Sir George Rekubrobe. On behalf of Prof, I'd like to say a very big thank you for this great honor, and I'd like to borrow the words of the German to say Dankeschön. Thank you.
Thank you. Thank you so much, Professor Horstman. And on behalf of Professor Dr. Rekubrobe, I would like to also appreciate you. Thank you very much. I think we have seen how it all developed. A lot has happened in 25 years. And as we know, KCCR stands tall when it comes to research stories. So for 25 years, a lot of great research stories has gone on at KCCR. Now we're going to start a few of those. I'm going to invite on the stage Professor Jürgen May, as well as Professor Yao Edusa-Kodie. They've had a collaboration between BNITM and KNUST, and they're going to be talking about causes of fever, infectious diseases epidemiology at KCCR. A wonderful applause for Professor May and Professor Edusa-Kodie. I think the mics are two, so.
What we'll do is that you start. I'm going to sit down. When you come back.
Okay.
I think what we've decided to do is that Professor May will start the presentation and I'll follow in after him.
Yeah. Thank you, Sachs. So thank you for the kind introduction. In 2 years' time, we will have the 125 years anniversary celebration at Bernhard Nocht Institute for Tropical Medicine, and I learned a lot in the morning how to do that and how to organize that. It was really a great event, and we don't have a king anymore. Nevertheless, I take some inspiration with me, and I started to change my outfit. I found this shirt on the bed in the guest house, and Ingrid told, and this was made 1 year ago, and Ingrid told me she does not know if it fits anymore, but it fits. So Ingrid, this is a proof. So, and I'm very delighted to see so many faces that I have not seen for many years that have collaborated over the last 25 years. So it's really, I'm very grateful for that. So now we have this, okay. Yeah, KCCR, it is difficult to recognize for you. These are different people. Over the time, people have come and gone. KCCR remains. 25 years already, and I dug deep in my computer to find slides and photos, and Rolf has already mentioned it. It is difficult to find it because during this time, 2000, we didn't have all the mobile phones with us, and the old presentations, I don't know why, but very often pictures and figures just disappeared, and some presentations were not even able to open. And those in the room, the senior researchers, may remember the time of 2000 when we had these kind of presentations. Oops. This was the time when we had slide projectors and very odd designs. This is a deep blue design, and yeah, I remember well the sound and the hot air from these projectors, even the smell, and some accidents happened all the time. And so in this case, many people in the auditorium woke up. And when the KCCR started, the PCR was only a few years old, and the internet was just born, and in the northern hemisphere. So what I want to say, when KCCR started, the world was completely different, in particular in research. So these were the two photos, the two oldest photos from 2001, what I have found, and you have seen these photos partly, or the right one, I think, in Rolf's presentation, and both of them are linked to human genetics indeed. And you have heard from Rolf already, human genetics was a very important topic. Indeed, there was one essential innovation at Bernhard Nocht Institute in the years before 2000, and this was molecular biology. And this allowed to answer new research questions on human genetics, and Rolf was the brain behind it. He has developed this concept and has performed and organized it here in Kumasi together with the co-PI Chiriak Ben-Yega. And on the right-hand side, you see one of the many, many young doctors, and this is Daniel Ansong. I don't know if he is here now, but now he is the Dean of SMS, as you all know. So, yeah, and during this time, KCCR was still, at the beginning, was still at the old SMS building, and Thomas Kupper started very early to find a new place and organized a new building here on the right side with an old photo camera. And on the bottom, you can see what I have found a few days ago in a box. This is a 1,000 Ghana cedis note that was used during this time. So we started with the group to look for other risk factors of malaria, and this was tuberculosis, one of the main topics of the human genetic studies. But here we looked for other risk factors. We looked for human host risk factors, for the parasite factors, for vector factors, and today this might be called a human One Health approach. On the left-hand side, you can see Christian Meyer, who was the supervisor during this time when I was a young postdoc. So we looked for many epidemiological aspects as well: temporal variance, seasonal prevalence and incidence, precipitation and other environmental factors influencing the distribution and the risk for malaria, and also entomological factors together with Thomas Krupper. And here in the middle you can see Paul, and he represents the team, and I would like to say thank you very much to all the people who have supported these studies, and Paul has served KCCR since the beginning on, since 25 years. Oh, this is tricky. Yeah, in 2002 we started the first big clinical trial at KCCR. This was on intermittent preventive treatment in infants, IPTI, and we could show that 3 doses of sulfadoxine pyrimethamine have a positive effect on the risk of malaria. And this was a study in more than 1,000 children, and we had more than 20,000 follow-up contacts. And Samuel Adjé was very instrumental during this time. This study was performed in the Afitya-Sechere district, and Robin Koppe was the first postdoc from Germany to be here for the study for a year. And you can see here the green car. This is the upper picture that has also supported all these studies, and I think it is still in use. I have seen it on the parking place with about 1 million kilometers on the tachometer. Yeah, then it became clear that children, pediatric patients where there was a suspect of severe malaria very often have bacterial sepsis. So, and we could see that the highest mortality of malaria suspected cases is under those with sepsis, and this was Jennifer Evans, who was the PI of this study. He is a clinical pediatrician, and she was also involved in these human genetic studies, and this motivated us to look further in causes of severe illnesses, and this is one of the main causes of mortality in children under 5. And we have performed these studies partly together with 3 partner sites in sub-Saharan Africa, and this was only possible through support of DECIF, the German Center of Infection Research, and I'm very happy that Dirk Busch and Timo Jäger, Dirk Busch is the director of the board of DECIF, the chair, and that they are today. And we had to establish better diagnostics, robust diagnostics, not only at KCCR, but also in the hospitals where we have worked. This is a continuous support of DECIF for KCCR. We have performed several different studies on causes of severe febrile illnesses. Sachs, Professor Ado Sarkodie, who will talk later, joined the team, Alice Owusu-Dabo as well, who is now the Pro-Vice Chancellor, and Dr. Nimako Sapong was a study coordinator and doctor for many years in these studies, and the largest was the Fever Without Source study with 4,000 children admitted to the ward involved, and 1,200 children included in the study, where we have looked in much what the causes of disease was beyond malaria. And we could see that most important bacterial infections are and this is very different from what we know from US and Europe, where we find mostly gram-positive bacteria, so it's another treatment. So very important to know that here Salmonella are mostly the cause, and these are mostly non-typhoid Salmonella. And we could also see in the fever without source study that more than the half of the children had malaria, or they had at least malaria parasites in their blood. But they had also other symptom complexes like lower respiratory tract infections, pneumonia, urinary tract infections, gastrointestinal infections. So the question was this malaria or not? Other causes? Yes, it was malaria according to WHO, because the definition is fever with Plasmodium falciparum infection. But were these really the causes of the severe illness? No, this is the wrong direction. And we could see that this is dependent on the parasitemia. The lower parasitemia, the higher the probability of comorbidity and co-infections. So very often the children had parasitemia, but other causes of severe illness. Another aspect, another aspect was the resistances of Salmonella, and Denise Decker, who is also here, has established bacteriology at KCCR for these kind of studies, and she could show that 77% of the Salmonella in the blood of the children were multi-drug resistant, so resistant against many antibiotics. But the good news was that all children, at least during this time, were sensitive against ceftriaxone, so cephalosporins, and ciprofloxacins, the most important antibiotics. But we could see already some mutations, so start of antibiotic resistances. So, and 3 years ago we started with a bigger study on a malaria birth cohort on the immunity of against malaria and this independence of microbiota. And here the PIs are Umu Maiga Ascofari, who is also the study coordinator since many years, together with John Amwasi and Nicole Gilberger. And all these studies made possible the conduction of clinical trials, for example, the ASAP trial, who is a multicentric clinical trial on the treatment of malaria in children with a triple artemisinin combination therapy as an alternative in case of emergence of Plasmodium resistances, and the PATVAC trial, pediatric phase 1-2 trial on a vaccine against non-typhoid Salmonella, and, for example, the establishment of a clinical trial platform, an RCT, on early medication against severe causes of COVID-19. I could not mention all our collaborators. There were a lot, and I'm very grateful for the participation in these many studies. First of all, I would like to thank today the Commander-in-Chief, Richard Phillips, and also Ingrid Sobel for the support for many, many years. And I would like to thank all the others and also the funding organizations to support this work. And I would like to thank the KCCR team for the good spirit and the engagement. And first of all, I would like to thank the communities and the people in the community to participate trustfully in these epidemiological and clinical studies. So I hand over to Sachs. So we have to go out and then...
Thank you very much. I'll continue with the fever without source work. In the early 2000s, I had just finished my PhD at the London School of Hygiene and Tropical Medicine when I got an invitation from Jürgen and others to visit them at the BNITM. So I was there to give a talk about the work I did for my PhD work. Now, then they got to know I'll be coming down to Kumasi. So that's when we started some talks about our collaborations. Now, the interesting thing about the mine talk is going to be that everything was bettered in KCCR. The initial work was all bettered from KCCR. But along the line, other partners also came in, importantly, the International Vaccine Institute in South Korea. So Jürgen mentioned that Salmonella typhi, which causes typhoid fever, was one of the organisms which played a big role in fever in children. So then we took on the Salmonella typhi work. Sorry. Can you move it on from the slide? It's not going. So the work with typhoid began from some early studies that had been done in Agogo, which looked at typhoid fever among children. And importantly, the burden of disease was very high in these children. For age group 0 to 15 years, a high burden of disease of 120 cases per 100,000 person years of follow-up. Now, the threshold of the WHO to determine a high disease burden country is 100 cases per person years follow-up. But in this young population, this is what we had detected amongst them. Now, at that time, we wanted to really establish the disease burden, but there was no standardized way of determining this in the whole of Africa. So there was a consortium that was formed called the Typhoid Surveillance in Africa Program. And that included countries from Senegal, Burkina Faso, Ghana, Ethiopia, Sudan, some cities in Kenya, and then South Africa, where standardized ways of looking for typhoid fever and looking at the burden of disease was done. In the Agogo area where we worked, we were able to collect 2,751 blood cultures from people who had fever, and we were able to isolate 135 microorganisms, which was significant. Now, from this 175 blood samples, 30 of them had Salmonella typhi in them. That's about 17%. And importantly, we looked at the antimicrobial susceptibility pattern. And the three commonest antibiotics we used to use at that time, ampicillin, cotrimoxazole, and chloramphenicol, the resistance levels to Salmonella was very high, between 60 and 80%. Luckily, at that time, ceftriaxone and ciprofloxacin were still very useful in the treatment of Salmonella typhi. Another finding from this particular study, looking at the disease burden again, was that in the age group below 15 years, this was far above the WHO threshold of 100 per 100,000 person years of observation. In the Agogo area, it was 389. So some other enhanced surveillance for typhoid fever was done. And in this study called CETA, Severe Typhoid in Africa study, what we looked at was, apart from the disease burden, to look at other complications that the children came up with, including intestinal perforations, hemorrhages, meningitis, etc. We also looked at the host response to Salmonella typhi and carriage rates. And also importantly, looked at the non-typhoidal Salmonella, which were involved in invasive diseases. So that again, in that same area, for all ages, the incidence, the adjusted incidence was still high, 118 per 100,000 person years follow-up, which was still higher than the WHO threshold. But if you look at other childhood groups, it was even far, far higher than that. So we did establish here that the disease burden in that area was very high. Among the findings, again, in the TSAP work, we had seen that there was resistance against the most commonly used antimicrobials. But again, for this, we looked at specifically ciprofloxacin, okay, and looked at whether the mode of resistance could be transmitted between other organisms. And we found that there were elements or markers of that easy transmissibility of antimicrobial resistance in that population. So these studies had determined that there was a high disease burden in the Asante-Echim area. Antimicrobial resistance was increasing to almost all the antimicrobials. So what could be the next thing to do? Typically, in the control of typhoid fever, is either using using wash properties together with vaccine. About the same time, WHO was going through discussions of pre-qualifying some typhoid conjugate vaccines. So then the next bit was to launch into a trial of one of these typhoid conjugate vaccines to see whether a phase 3-4 trial in Ghanaian children will find the vaccine effective in these. So currently, the Tyviga work in Ghana here, which is ongoing in the Asante-Echim North District, is looking at the effectiveness of a typhoid conjugate vaccine in children 9 months to 15 years old. We have already vaccinated 20,000 plus children. The vaccination is finished, and we are following them up for the next 2 years. Last week, we had a stakeholders meeting in Accra because the government has started talking about the possibility of a typhoid conjugate vaccine. There's an 85 million grant from what do you call them? From Gavi for any country which is interested in using these vaccines. But we need to generate data for them. So we had a stakeholders meeting in Accra with about 80 people, the World Health Organization, Ministry of Health, Ghana Health Service, other CSOs, to go through our data, which could support the policy for the introduction of a typhoid conjugate vaccine in Ghana. We have been interacting with the National Immunization Technical Advisory Group, who wanted information on these things so they could talk about the vaccine. As I've said previously, our threshold in Ghana is high. In fact, in West Africa, it's Ghana and Burkina Faso. Incidences of above 100,000 used to be seen in Southeast Asia. But in West Africa, at least for Ghana and Burkina Faso, we have seen that the incidences or the burden of the incidence of the salmonella typhi disease is very high. So through the work we have done, which began in KCCR and continued later with some support from the IVN, we have determined that the incidence of disease is very high in the country, significant mortality and morbidity as well, and complications from typhoid fever. Importantly, there's multi-drug resistance in the area, and also that there's evidence of transmissible drug resistance in that area. We looked at the literature compared to other African countries where typhoid conjugate vaccines are being given, and other parts of Asia as well, which all spoke for the safety of the vaccine. We are still, the trial is still ongoing. Okay, we haven't finished yet, so we have not broken any code to find out the safety, but we are pretty confident. After the vaccine we followed down for quite some time, and we're happy with the kind of adverse events the children are coming up with, which are nothing to write home about. It's also been determined in other places that you can't give this vaccine as low as children who are 6 months. So this could fit in very well with the expanded program of immunization. One of the things the KCCR director talked about earlier was the issue of capacity building. That within all the work, within this typhoid work, we've built in some training schedules for our students. Some have completed their PhDs, other PhDs are ongoing. So this is what, within the fever work, has been done so far as typhoid fever is concerned. And on those days, as KCCR celebrates its 25th anniversary, all we can say is congratulations for the support, for the initiation, the birthing of this typhoid work in KCCR. It is my hope that the next 50 years ahead, we'll see you moving on. Thank you.
Thank you so much for the wonderful presentation. That's Professor Yagan May and Professor Yao Edusakode. A round of applause for them one more time. Well, our next one is also a collaboration, but this one is between Charité and KNUST. Please help me welcome Professor Christian Johnston and Dr. Augustine Silverkin. But I think I'm going to let Dr. Silverkin sit through a little. When he's halfway, then you can come in. Please, a round of applause for Professor Christian Johnston.
Yes, thank you. Yeah, it's great to have such an event here for such an occasion. The contribution that I'm going to present is probably not comparable in its extent in the portfolio of KCCR, but still, I think this is somehow a foundation to what's been mentioned all over the place in the ceremony today, which is the response to COVID-19 provided by KCCR. So this actually is the groundwork for it. The work on, let's say, pandemic or emerging viruses done mainly in the beginning together with SACS, so Professor Edusakode, who just spoke, and then Augustina as the first, how should we say, postdoc, right? You were a postdoc on the first grant. And we had a whole series of projects in this regard, and I think it shows also that KCCR is able to mediate contact with departments of the university that are not medicine, right? So we work with the Natural and Renewable Resources Department and so on, brought in zoologists from Germany, so something a little bit out of the norm, but still interesting. So what we have been working on for almost 15 years now is concepts like this, the species barrier, right? So a virus coming from a natural reservoir, from animals, and then switching a species barrier, acquiring the potential to cause sporadic cases in humans, and then also onward adaptation in humans, adaptation for human-to-human transmissibility. So when we conceived this work, we knew little, right? We were just clinical virologists, and what we knew after SARS, this was our stimulus actually, was there is host animals like these, known in China as hosts of SARS-1, so the initial SARS coronavirus. These are carnivores, and we did a lot of work on species like this, also here in Ghana, but they are quite difficult to catch. This was more the subject of the second funding period. By the way, this is how probably this livestock industry in China was stimulated and expanded. There is a lot of money to be gained. What you see here is raccoon dog fur, and this raccoon dog fur is almost exclusively produced in China for the world market of garments. And well, you see where these pieces end up, right? You know, these pieces of garment has become fashionable and acceptable again to have small pieces of fur. This is, well, probably the result of a pandemic. Also, we knew Ebola virus, a topic that also then later on became quite, well, up to date here in Ghana. So the first big capacity building activity after our research-based activities was then the Ebola virus training and testing project that came up. Well, also for Ebola virus, it was clear there is a natural reservoir. These are fruit bats. Here a story by Eric Leroy describing this in Gabon. And the institute in Hamburg had been active on Ebola ever since. Herbert Schmitz as the Department of Virology, but then also people on the ground like this picture taken from the 2000 Gulu outbreak, Ebola Sudan virus outbreak. People like Sabine Mand, Matthias Grade, Hinrich Sudeck, these names are well known to Hamburg people. They were there to provide outbreak assistance. Yeah, so we somehow naively started to catch any bats around, right? So this, for instance, is a hammer-headed fruit bat catched in the botanical gardens on KCCR campus. So these guys are flying around here. These are, well, they live in small groups, small flocks. They form harem-like structures, and they have a very typical call sound that you will know when you've stayed here at the guest house. You hear them in the late evening. These are big animals, like, well, more than a big bottle of beer when you hold them in your hands, right? These are quite big animals. But they do not host SARS-related viruses. They are more like relevant for filoviruses, although we've never found filoviruses in these animals here on campus. This is another species, an Epomops, looking like a little dog. They were more comfortable when we hold them this way, right? Because that's what they are somehow used to. And there is also a big colony of eidolons in the botanical gardens that many of you will know. They are also served in this form. You can have them with and without hot pepper sauce from a barbecue. This is actually a picture taken by one of the PhD students from Ulm Department of Zoology, Heather Baldwin, and I think Michael Owusu was also around when this picture was taken, right? So that's during the field work we did in villages, trying to find out what actually are contact activities and how does the contact zone between humans and these remote animals look like. So there is things like sociological investigation associated with this type of study. That's interesting. So interdisciplinary research can really be done in this environment. These are the same animals alive. That's a colony roosting in the botanical gardens, between 300,000 and 400,000 animals. They breed, they come in here to breed, and also these animals we studied, and there's now a new project coming up, a short-term capacity building project, sequencing the leftover samples from this project, and by doing so, providing training in next generation sequencing. Most of the team is here in the room, most people. Several others will come, and the project will actually take off properly in January, and will run only half a year, but it somehow shows what this type of work has afforded all of the time: capacity building, right? So the capacity to do virus detection, this is the main spin-off from this activity. Yeah, here you see them flying off to feed. By the way, this colony sitting in the botanical gardens, it consumes 4 tons of fruit every night. I don't know where they find them, but they have to eat this much. And then, of course, there is the interface, right? This is the central market, and the animals are flying right over the head of these people, and they are leaving their droppings there. And we know these droppings are full of paramyxoviruses, some of them related to Nipah and Hendra viruses. They are sequence characterized, but we know a little more about these viruses, only that they are around and probably people sometimes catch them. This is how we did the sampling there. So this is just plastic foil from the construction market, bricolage market, and you just collect what's dropping there and put this in PCR tests. Yeah, you can isolate viruses. At that time, we had cell culture established here at KCCR, and I think we should re-establish this again because there is a new P3 lab. So this whole capacity building aspect is is important, and this shows that you can really do virus studies. By the way, Tabea Binger was shown in the morning when she commented on the coronavirus testing effort here. The German head of labs, she gave an interview. She did her PhD on this project, so she stayed here for long periods of time at KCCR. Yeah, we know this virus is transmitted, so the fruit bats have it at a seroprevalence about 50%, but also swine have it at 30% seroprevalence, humans at 11% when they are exposed, which means when they are working at the zoological gardens. Non-exposed people have much less antibodies. So these are interesting studies that are not even concluded. And we did field work in mainly these places, Kwamang, or little workarounds to do that. So this is a more detailed map of the sampling sites. Actually, we explored the whole country in the first funding period of the DFG project for bats, but then also going to livestock. So the little yellow dots here you see, these are more livestock sampling sites, but also bats were sampled here to have, well, outlying sampling sites. And what you see is viruses related to human pathogenic coronaviruses in bats, but the relationships are quite distant here. Then a periodicity, when do we find, which time in the year do we find actually virus-positive bat droppings? And there is a clear periodicity, which is not climate-associated, but parturition associated. So they have a parturition peak in the year, and that's actually short after when there is virus amplification in these bat groups. These are caves, so mainly most of this is cave-dwelling bats. They look different, they are smaller. Another interesting spin-off also is cell cultures from these animals. So while here at KCCR we made probably close to 100 cell cultures from bats, from mainly the lung, some of them epithelial, so we are still working, and infectious diseases researchers across the globe actually have received cell lines from us and are working on this. So a lot of good, let's say, Ebola virus research in the US is carried out nowadays on cell lines we made in Kumasi. So quite an interesting and important contribution that's often not really recognized. Okay, yeah, so there is a wider field which is, has become a buzzword. I still like to call it zoonotic diseases research or disease ecology, but nowadays you have to say One Health. You do One Health research. Okay, so we also do it, and KCCR can do it as well, right? So all of this is One Health research and has already happened here. So there is now work going on poultry. I think the Friedrich Löffler Institute has been involved now. There is work on swine. Our project has done a lot of work on swine hepatitis E virus with a whole number of independent publications from KCCR. We are now moving somewhat further on to animals we don't find in Ghana, to camels. So we are working on MERS. And also here it is interesting. So there was the belief that actually this disease doesn't exist in Africa, but as soon as you start looking, you find it. This is a whole number of sera we collected in Kenya and studied there, and we see neutralizing antibodies in about 2 of 1,000 people, and this is the typical prevalence we also find in Saudi Arabia. It's the same, so it's not more or less in Africa. Okay, in Ghana, in the bat studies, of course, there was a lot of linked questions. So it is quite obvious now, we know by now that these viruses are not transmitted all over the place to humans. This seems to happen very rarely, but still we did studies asking how and where people have contact with these bats. And it's surprising, but in remote villages, these bats are really a source, for instance, of income, because they take fertilizer from the caves. Also, they are a constant source of nutrition in some settings, and they encounter bats in their everyday life. For instance, in one of these villages, the school was infested with bats, and all the young school pupil They know they see bats every time they go to school. They have to somehow chase the bats away. Then there was another department in the project, Michael Owusu. He went to the villages to test for human common cold coronaviruses, and these are actually the only data available showing that in remote populations, remote groups of people in Africa and in villages, actually these viruses behave like they behave in industrialized countries. So these are also common cold viruses. Also here, the pronunciation of symptomatic disease is with children rather than adults, because adults are immune. These are endemic viruses, unlike SARS-2, where this is different. And also here we see that in fact there is a close relationship between people being symptomatic and shedding a lot of virus. So people who are symptomatic shed a lot of virus, and these are more likely to be children. Okay, so the second period then sampled livestock across the country. So I don't know how many, but I think the number is around 3,000 pieces of livestock, mainly goats and cattle and swine were sampled. And well, we also here managed to establish good diagnostic work now in the Faculty of Veterinary Sciences, which is another structural component I think the project contributed. So a proven collaboration also on research subjects with the Faculty of Veterinary Medicine is provided here. And we had several people who came on stipends to be trained in Berlin and went back, and people like Vitus, for instance, they established immunofluorescence testing at KCCR for these projects, the sampling through the Department of Veterinary Medicine, then the lab testing conducted here, and seroprevalence studies in cattle entirely done in Ghana. Not a single sample tested in Germany, which I think is a merit worth mentioning for the place. I think it's important that you can really prove this is not only theoretically possible, but it has already been done, and we've already published about it, right? So with this, I thank KCCR and the university. These are the main collaborators. Sachs, then for the social sciences part, Olivia Agbenyega. I think she was here in the morning at least. I don't know whether Olivia is still around. And then, sorry. The colleagues from the veterinary medicine department, Sam Oppong here. Sorry, I'm really a bit tired. Sam Oppong. And then Folice, yeah, of course, Raphael Folice, and then his successor, Ben Emikbe. Yeah, sorry, I'm really a bit tired currently. Yeah, and the students. So most of them are here currently. You see them here in field work. This is during the animal sampling part, and you see how they had fun in the field over these many years of work. So I thank all of you, and I thank KCCR for providing the infrastructure and the opportunities and these great people and great collaborators we found. Thank you.
Well, thank you so much, Professor Christian Drosten. Dr. Silverkin. Okay, thank you. All right, all right. And that was virus emergence and emerging viruses. All right, so moving on, we have a few to cover, and two more, because all these are very important research that's going on at KCCR. Now, the next one is 25 years of filariasis research at KCCR. I'm going to invite Professor Hakeem Horof. A round of applause for him. Is Professor Deborah helping too? You're going to, oh, you come after him. Okay, so Prof, you're going to start and then he'll join you soon. A round of applause, ladies and gentlemen.
Yeah, thank you very much. I really very much enjoyed first being invited to this great event and also to see and be part of this great celebration tomorrow. I don't have an anniversary to celebrate like Jürgen in a few years, but I would also take something home from the way how you celebrate and how you really make this a big event. Now the first slide is on. My task is, and Alex Debra's task will be to squeeze 25 years of filariasis research in a few minutes, which of course will not be easy, but okay, I will hopefully not shoot it off. It will also hopefully come clear how difficult sometimes it can be to really run projects for such a long time, because they are in principle on third-party funds and therefore really not per se sustainable. So you always have to actively sustain them. But we were lucky to really continue with a series of grants, so to stay in this field, which still is very interesting, but where over the long period you really can see that there is now movement towards paradigm shifts in drugs and so on to really end filariasis maybe in the next 10 years in the different countries in Africa. So there is a lot of funders. Usually as a postdoc you start with small grants by DFG, and we were lucky that DFG decided to have special foci on Africa projects to accommodate the special needs like logistics and others. And this helped a lot, also helped the European Union and Volkswagen Stiftung, so that you could, like a mountaineer who puts in a hook and then climbs the next step, really rely on one after the other project. And in the end, I was very happy that at the occasion of the 20 years anniversary of our joint projects that started in Hamburg and are now continuing from Bonn, that the University of Bonn was enough interested to sign a memorandum of understanding. So here you have our then Vice Chancellor for International Affairs. He is actually a historian and has worked a lot about migration from Africa into the US and so on. And they signed a memorandum of understanding, and our international bureau, so Christine Müller is also part of the entourage this time. She helped a lot in making it real that we got Erasmus exchange programs so we can intensify the student and also professional exchange. So we, as I said, we still work with diseases that when I started were said, they are going to be eliminated in 3 years. Would you really want to engage on them? But in the end, it has proven that it was a good decision to stay, because there are still several nuts to crack. And one, for example, I'm going to start with is the development of new drugs that would really bring about the possibility to end onchocerca and lymphatic filariasis. And the other will be that methods and tools be developed for the disease sequelae. For example, lymphatic filariasis you can eliminate with regard to transmission, but people who have the disease, because it's becoming independent from infection, they will stay with thick legs, with elephantiasis, with hydrocele, and you have to do something. And this is also something where you can really bring in modern tools of access, of participation, of country ownership, and all these things. I will start with the drug development, and later Alex Debra will go to the morbidity management. I'm a parasitologist, and so I have to show you transmission cycles, but this cycle serves the purpose to understand that, for example, in onco and similar in lymphatic filariasis, the problem are the long-lived adult worms. So we do have drugs that kill the larvae and interfere with transmission, but the adult worms keep producing offspring, and therefore you have to extend these programs for more than a decade normally. And if the coverage rate in the communities that you're targeting is not high enough, you have to extend. And so projections go depending on the efficacy of the local health systems, that some countries may eliminate filariasis in the next years, others will have them around for the next 10, 20 years or more. And even though when elimination is reached, this is always a threshold. So you will still have individual disease popping up here and there. And what you need is curative drugs. And this is also a necessity arising from the Sustainable Development Goals of the UN, which ask in Goal 3.8, Universal Health Coverage, that there are curative drugs for the people, and they should not be paid out of pocket, so there should be health systems paying for it. This is basically what all countries who signed the charter have agreed to, and I think it's becoming clear that this is a huge task. So anyway, back in the Bernhard Nocht Institute, Dietrich Büttner, the late Dietrich Büttner, who was one of my mentors, the contract was made, group leaders and postdocs would present. And I remember it was in Bernhard's office where we were sitting there, Robbie asked me, "You are a medical doctor, why do you keep working with mice? You should come to Ghana and do a human trial." And I was sort of surprised how easy it sounded, and then we set out with the senior experts who had already some experience in trial to design one, and it became clear that what worked in mice also worked with Onchocerca in humans. So this is a normal Onchocerca worm in a nodule, transverse section, and all these little red dots are the bacterial endosymbionts, the Wolbachia. And if you treat patients with doxycycline, then they are depleted, and what you can also see is that the uteri here, they are empty from embryos, and we also found out that the worms do not live 10 to 15 years like they normally do, but they die within a year. So it is a slowly working, but a macrophyllaricidal or adulticidal principle, and the slow activity is actually beneficial, because if you have a fast worm killing like you would have with suramin if you took it, you would have a lot of side effects because of the worm death, which is not seen after the slow death. So following this initial discovery, we did a lot of fine-tuning studies, tuned down the dose and the time, and we extended to other diseases like other filaria, like lymphatic infection and also Manzanella. And unfortunately, you may not be able to read it in the back rows, but in the first rows, you may realize that the first authorships very quickly took over from German authors to Ghanaian authors, mostly by the name of Deborah, but also others as the studies moved along. So we started the principle and could uphold it until now, that we really share project management, project design, and of course also publications. Now, the doxycycline treatment is part of, if you wish, general medicine. Also, it's an orphan indication, so it's not registered. It would never find enough money to register it, but it's being used widely. It's also being widely used in veterinary medicine, by the way, for dogs, because heartworm is also a difficult disease. When a dog has heartworm, the fast-acting adulticidal drugs would, in 10% or more, also kill the dogs. So it's better to use doxycycline. And I was pleased to read a couple of years ago, I knew that it was coming, that WHO through PAHO, the Pan American Health Organization, adopted doxycycline for mop-up and endgame strategies in Southern America. I hope that the political crisis in Venezuela will not prevent success. Let's see how it works. But it is clear that doxycycline per se, because of the long treatment and the contraindications, is not the ideal drug, also it's used. And therefore, the Gates Foundation 15 years ago decided that knowing the mode of action of doxycycline, they could set up larger drug screening studies, because it's possible through an insect cell line to do high throughput screening. And this was the work of then Liverpool School director David Molyneux, who also invented the term NTDs, to convince the Gates Foundation that this is a principle with which they could start a macrophylloricidal program, which then extended also beyond Wolbachia. But this is essentially how it goes. You treat patients, and then you operate the nodules. We did that last week and did some more movies. Actually, I could have put them in there, but I can show those interested at another time. And these nodules will then have to be analyzed for drug efficacy. If you want to show a macrophylloricidal effect, you need to show that the worms are actually dead. And this is now the task. So this decade of Gates funding was not without success. It was a big consortium spread out over the world. Many labs contributed to that, and we have now a few of these drugs indeed in phase 2. One phase 2 is taking place in Ghana with emodepside by Bayer. Another one with AbbVie, it's an anti-Wolbachia drug. The anti-Wolbachias are in blue, and the direct acting ones that are also coming along are in red. So this one study is taking place in the Volta region in Ghana, and the AbbVie study, both sponsored by DNDi, by Drugs for Neglected Disease Initiative, are from Congo now. And there are two more candidates, one at Liverpool, an anti-Wolbachia, and a DCIF drug, corallopyronin, which actually, because we did all the animal studies, shows the best adulticidal efficacy. So we'll see how the other candidates behave. There will still probably be some attrition during the clinical phase. One important thing that came up when DNDi first spoke with the regulators for registering these studies is how you parameterize the anti-adult worm efficacy. And the usual answer is there are a few experts in the world. Notably, they all come from the Hamburg lab. They were all trained by the late Dietrich Büttner. Some are in St. Louis now, the others are in Bonn. And this was not enough for the regulators. So they said, it's nice that you have good experts, but how would you really want to nail this down? And therefore, they called for machine learning, for artificial intelligence. And this is obviously, again, something what the Gates Foundation loves, to mechanize everything. And so, and it's really complicated. Doing the same mistake. It's really complicated if you have big nodules with a lot of worms. So how do you assess that? But it's, on the other hand, it's interesting to see we got funding then for an artificial intelligence project, and we teamed up with the international company Capgemini, who has a big German headquarter in Cologne, but worldwide, I think, 100,000 employees, and a lot of them can contribute by tournaments. So they have a very nice way of getting the energy from different parts of the world. So it is possible, it has been possible to really arrive at a degree of agreement between human assessors and the machine within half a year of over 95%. So we are now in the process of prospectively evaluating that with the incoming studies. And the idea is that for registration, there will be only one human assessor doing like 10%, and the rest is done by an algorithm that we have to put down as part of the registration process of these trials. And these are a lot of people that are involved. So there is DNDi, my former colleague Sabine Specht is now there, head of clinical filariasis. The former Büttner trainees, Peter and Kerstin Fischer, are now in Washington University are taking part, Capgemini, and of course the team in Ghana providing the samples. So we hope that we deliver a tool in the next 1 or 2 years that is now fit for registration. Actually, I took some of the contents already, so I can go over this slide. So what the machine has to learn is to find out what is a worm section and what is crab, What is easy is to quantify the Wolbachia, much easier than for the human eye. The sex and embryogenesis seems also relatively easy. I was surprised how quickly the machine could actually learn and come to similar conclusions with the human assessors. So it should be hopefully developed as a tool over the next 1 or 2 years. I started in the Department of Immunology, and of course my heartbeat was also for parasite immunology, but due to time constraints, I just do a summary slide here on things that we found out over the years. So one was to sort of negate the idea that protection against worms is Th1 mediated. It's very clear that the protection from animal models and also from human studies is Th2 with some, if it comes to tissue, Th17 dependent immunity. And this also extends to the serological parameters. So IgG4 is usually the subclass that is produced by regulatory T-cell responses and tunes down the pro-inflammatory responses. So very clearly seen in lymphatic filariasis where, for example, people with disease have clearly lower IgE antibodies and higher IgE antibodies than the ones without clinical symptoms, where probably IgG4, maybe even pre-birth already tunes down the immune system. Now, I go on icy terrain since Christian Drosten is here talking about COVID, but we were appalled by the, I mean, a lot of our studies to start with had to deal with the fact that worms immunomodulate, downmodulate immunity. And when it became clear that in addition to, of course, the population being younger in Africa, oops, it should, yeah, it shouldn't go off. There was still surprisingly fewer, and thankfully, fewer severe cases that one would have maybe feared. Other groups hypothesized already back in 2020 that this may have something to do with the co-infection, so that the balance of a dampened immune response might actually be helpful preventing severe COVID courses. And this may even be also due as a second co-infection to malaria. This may, if that's true, it would be maybe even the stronger driver. And if you compare the maps, severe COVID, severe malaria, they are really pretty much complementary. So Thomas Borjomime, former DAD student, now postdoc in my lab, he tried a little bit COVID immunology, and he could see that if you stimulate T cells from human reconvalescents with the S protein peptides, there is some degree of activation of T cells, of CD4 cells, more actually by CD8 cells. But if you then co-incubate with human, with helminth antigens, so Oncocerca or Brucia antigens, the activation induced by COVID peptides goes down in CD4 cells, not in CD8 cells. And more interestingly to me, the pro-inflammatory cytokines, TNF and interferon, are strongly reduced and IL-10 is built up. So this may contribute to a certain extent to more moderate courses in COVID and maybe prevent one or the other severe course. A lot of work was also invested to find out why a few % of people after lymphatic filariasis actually acquire lymphedema, and this clusters in families. So obviously this is something that calls for genetic analysis. We also discussed still in Hamburg with Rolf, and then went on to in discussions with my Bonn colleague, Markus Nöthen, and started first to do some good guess studies of SNPs that we thought could be involved. It should have something to do with vascularization, and indeed, for example, matrix metalloprotein or CCAM, that is also lymph angiogenesis factor, and directly the vascular endothelial growth factor, SNPs, came out significant in that study. And interestingly, this held also true, I cannot go into the details, on the protein level, allowing Linda Batzer-Deborah to draw this nice network of proteins that could lead to lymphedema propensity in families. And with that, as Vorarbeiten, as preliminary work, can far help me in designing a genome-wide association study together with Alex Debra, where we really recruited a high number of patients and analyzed an even higher number of SNPs. And there is at least one that makes it over the -10 to the -8, and also therefore should hold a multiple million Bonferroni correction. And this is again a gene that has something to do with interstitial fluid, so it might have been something functional to do with lymphedema. Sorry, I was probably dying down with my voice, as I realize now. Yeah, and as in the other projects, Christian and Jürgen already mentioned there, of course it allowed us to train a lot of people, and the nice thing here is if you really go through a PhD, you have very good chances of making your career in academia. And good examples, I mean, first of all, Professor Alex Debra, who started as a master's student and now has been even dean of a faculty here, but also Linda Batzer, Jubin Osim-Mensah have become lecturers at KNUST after their PhD, and a lot of other masters made it also to interesting careers. And I think it's very important to have PhD programs running, because then these new experts help you in the collaborative effort. And again, also in genetics, a lot of papers with Ghanaian first authors, of course. Here is the point where I hand over to Alex, because the BMBF research has made it mandatory that the joint collaboration is actually managed as a PI, not by a German, but by an African colleague. So I'm in this project the co-PI, and so I hand over to my PI, Alex.
Thank you very much, Akim.
This is forward, backwards.
So the good collaboration between Germany and KCCR continues. And here, as you can see, the TEK-OFF consortium is actually a consortium between 5 centers, 3 from Africa, as you can see, from Tanzania, from Cameroon, and from Ghana, and then 2 partners from Bonn and then Munich. And as Akim already said, I'm the overall coordinator, and Akim is my co-coordinator. And it's one of the 5 research networks for health innovation, actually sponsored by both German Ministry of Education and Research. And so far, we started with 2.8 million euros, but at the end of 2022, we've been given over 3 million euros to do this work. And the main partners, as I said, you can see is from Cameroon, Tanzania, and Ghana. And the main aim of this consortium work is to actually do mobility management and treatment of filariae early, and then pseudoconiosis. And then we also did a bit of digital work, which I will show. So how did it start? The global program to eliminate lymphatic filariasis has 2 main goals. The first goal is to actually stop transmission, and then the second is to actually find treatment for those who suffer from the pathology of the disease. The first one has been successful. Through mass drug administration, LF has been eliminated from many countries, and if you take Ghana, for instance, out of the initial 98 districts which were endemic for LF, now we have less than 10. And even in these 10, they haven't passed TAS, that's transmission assessment, survey. But when we went to do survey, virtually there's no transmission going on, which means the first aim had been hugely successful. However, when it comes to morbidity management, it has lagged behind. So as a consortium, we thought of what we could do to actually help. So it all started in 2012 when we recruited some lymphedema patients and gave them treatment, that doxycycline and amoxicillin for 6 weeks. And we saw an improvement in those who took doxycycline. As you can see, this is an elephantiasis patient or lymphedema patient with stage 3. And 2 years after treatment, the stage moved from 3 up to stage 1, which is clear evidence that doxycycline has some effect in ameliorating lymphedema. So based on that, well, as Hakim said, when you look at the Sustainable Development Goal 3, specifically 3.2, it says universal health coverage for everybody and no out-of-pocket payment for NTDs, which means the government or agency should find money to actually treat these patients. So based on the previous results that I've shown, we came together as a consortium, 3 from Africa. I mean, on LF, we actually had 2, 1 from Ghana, because we were the pioneers who actually did that. And then we had Tanzania, which came on board. And then we had also Cameroon, but Cameroon didn't work on filariasis, lymphedema or filaria or regima, they work on podoconiosis. And then we had 3 other countries, that's Mali, Sri Lanka, and India, which were organized by task force. They were sponsored by USAID. So we joined forces together in these 6 countries, and then we developed a common protocol to actually try doxycycline, for 6 weeks. So as you can see, these are the centers. Well, the summary of the results is that after 2 years of treatment, we didn't see progression in 98% of the study participants. Actually, there wasn't any difference between doxycycline and placebo, but 98% did not see progression in their legs, which means the treatment was able to actually halt progression of the disease. And in 22% in both doxy and placebo group, actually we saw improvements. And here the question is, why is it that the first work that you did, you saw doxycycline was effective, but in the second and then the multi-country trial, we didn't see effects? We think it's because in 2006, when we did this, the first work, transmission was very, very high. In all the districts where we did the work, transmission was very high. And when we did the second work in 2017, 2018, the district where we did the work, transmission was virtually 0. So because transmission was not going on, there was no introduction of new early into the patients, and we think that's why maybe we didn't see any effect from doxy. The second one was that in the first one, the hygiene training or hygiene measurement wasn't the best. But in the second one, we actually followed the patients religiously. Every month we were in the communities, we were giving them hygiene material, they were cleaning their legs, and you could see that hygiene was very, very effective. So we think probably because the hygiene was very effective, that's why we didn't see any extra effects from doxycycline. However, it is good that now, because all along WHO have been, recommending hygiene without any proof or evidence. But now we have proof from 6 centers that yes, hygiene is effective in ameliorating lymphedema. Again, one thing that we saw was quality of life. As you can see, this is disability score from Ghana at the beginning. Quality of life was, as you can see, is very high here, which means the standard of the quality of life was very low. The people didn't have good quality of life. But 24 months after treatment, we could see that there was an improvement in the quality of life, which means, for the patients, they know the size of the leg might not reduce to the normal, but what they need is quality of life, that they can go to their farms, they can go for fishing, whatever they want to do, they could do. But before then, they were bedridden and were dependent on their families. But after treatment, they could do whatever they wanted to do, majority of them. So what we also did was, as I said, hygiene training for all the patients. If I can show this video. As you can see, the top one was from Anna, basically from Katsina and Canal District, that's KCCR pediatric staff teaching lymphedema patients how to wash the legs. And this is from Tanzania. And at the end of the day, you could see that all the patients were able to actually do the washing themselves. And you can see here that, you know, the hygiene data showing the percentage with clean limbs reflections in compliance to trained procedure that we were showing them. So what is the interpretation of our results and the next steps? That's the takeoff two. Here you can see that an unexpected large positive clinical outcome in all patient groups likely resulted from the repeated training and close attention to MMDP that we gave the patients, as I've already explained. That it is likely that because of the intense and the religious nature that they follow the hygiene, that's why we couldn't see any difference between doxycycline and placebo. It doesn't mean doxycycline is not effective. Secondly, a follow-up study of this same closely followed individual will address a major current WHO conundrum. Here I have to say that from the good work that we had from the first, that's the first takeoff work, we were able to, we succeeded in getting takeoff 2 out of, I think how many, about 18 applications, only 5 or 6 were selected, including takeoff 2. And from the old consortium, out of 5 old consortium, only 2 were selected, including takeoff. So we were happy to actually receive this, which will give KCCR extra 2 million euros for 5 years. And for this, what we are going to do is we are going to follow up on these old patients, try to see why is it that even though food hygiene is effective, yet the patients are not able to follow that. And then we will employ the social science aspects, and we will include, as I said, we include all the old patients that we excluded, because we wanted to know why those patients that we excluded some patients from the treatment. And when we went back 2 years after treatment, we saw that those who were excluded from the trial, they didn't have improvement in their legs. In fact, their condition got worse. So the question is, we taught them how to wash their legs like those in the study. Yet 2 years after treatment, how come that those who were in the study, they had improvement in their legs, but those who were not included in the study did not have any improvement? Even though if you ask them, they said they were also washing the legs like those who were part of the study. So for this second phase, we are going to find out why those who were not in the study did not have improvement, but those who were in the study had it. Again, this is another part, as you can see. 3 years after treatment, when we asked the patients, as you can see, about that's 259 in group A. They had reduced in attack. And group B, out of 60, 41 had reduced in attack. So as I said, for the patients, what bothers them much is the attack that they get. Periodically, they get attack of the legs. When they get attacked, they are bedridden. They are not able to go to their farms. They are not able to do anything. So for them, if they get relief from the attacks, they are very, very happy and grateful. And as you can see, 259 out of 3-something, they had reduction in the number of attacks per year. And here, out of 60, 41 had reduction in attacks. One of our flagship web packages was the use of mobile phone, actually the interactive voice message to get patients. Now the traditional method is when they are doing MDA, the community health workers go around, list the people, and then report to Ghana Health Service how many people have lymphedema. But we thought in this part of the world where illiteracy is very high, the community health workers are not able to report properly. So through Takeoff 1, Dr. Linda and co developed a system using the mobile phone, actually the voice messaging, because the people could not actually, majority of the people could not text. So we use the voice messaging. And as you can see, the algorithm, you are not supposed to actually know everything, but just to tell you that through this algorithm, the community health workers who live with the patients are able to report if they get new cases or old cases. They are able to report into the system, and wherever you have, you're able to see that a new case or an old case has been identified. And from this work, we were able to see that this system is able to identify more cases, 10 times more than the traditional method, which is understandable because here they have to call into the system, or like the traditional method where they have to write. And this is very important because according to the WHO, for you to be certified to have eliminated LF, you need to put measures in place. You need to find a way of identifying cases. And we think through this system, almost every patient in remote areas will be able to be identified. And then the second part is those who have attack. The community health workers are able to identify them and report into the system. So if you have the system in the district or in the CHIP compound, if a patient has an attack, this patient will report into the system, and then the health center or the clinic will know that there is an attack here, and they can follow up on this patient and give that patient relief. And we think this is very, very important for the patient and also for Ghana Health Service. So as Hakim said, we've trained so far 6 PhD graduates. Dr. Linda is with medical school. Myself, allied, Dr. Jobin with School of Veterinary Medicine, Kwame Kwatu with College of Science, and then Dr. Labi with Department of Biological Science, and Dr. Ebu Amafo with the sister university. And I think 5 of us were funded by DAD. And again, as you can see, this is the funding actually coming to KCCR through the projects. So far, Filari's project has brought over 10 million euros in the past 25 years to KCCR. Again, you see intensive collaboration going on here. This is a picture which was taken in April when myself and Dr. Linda were there. And as you can see, we have 2 PhD students and 2 master students in Akim's lab. And then, before then, we had a PhD student from Germany who actually came to Ghana to collect samples. And then, interestingly, 4 months later, when he came to Ghana, these were the people he worked with. And 4 months later, they were working in the same lab in Akim's lab. So far, from Bonn to Ghana, we have 10 students coming to Ghana. And from Ghana to Bonn, we have 15 students, both masters and PhD. And for staff exchange, from Bonn to Ghana, we have 10 staff. And from Ghana to Bonn, we have 10 staff. And these are the funding agencies, DAD, Erasmus, GWAC, etc. Actually, we've moved from the field to Memorandum of Understanding. As Akim said, KNUST-TEK has actually formed MOA with Bonn University. That's where we're able to exchange more students. And then here you can see Akim in the field, very tired. That's the essence of collaboration because they don't just sit in Bonn and then tell us bring the sample. They come down, we all go to the field, and then where there's the need to actually go to Germany to do some lab work, we also go there to do it. Again, here you see Akim, myself, and Dr. Linda explaining the procedure to the patients. And we've actually moved from the old system where when you want to do a clinical trial, you just put forward about 16 to 20 page application. And now we do it, by good clinical practice. And as you can see, Akim holding. Now we developed 262-page protocol, which is actually monitored by Food and Drug Authority. They always come around to see that we are doing the right thing. For NTD advocacy, Angela Merkel has pledged, she pledged her support for NTD before leaving office, and you can see the NTC gurus actually discussing the way forward for NTD. And this is Akim with the president. Actually, he was hoping that today the president will come so that he can meet him for the second time. Here they met in Düsseldorf, I think. That's when the president of Ghana went to North Rhine-Westphalia for partnership. And I didn't know what they were discussing, but you can see, I can imagine the president promising Akim that he will help eliminate NTD. So we'd like to acknowledge our partners, as you can see from Germany. That's Prof. Butan of blessed memory, who actually trained Akim and myself. And then our study partners from Germany, Ghana, and more especially to our patients. This is a patient that we interacted with last week, Akim and myself, because BBC wanted to interview the patient on the importance of ONCO, how our treatment is affecting his life, and then the artificial intelligence model. So we thank all our sponsors and our collaborators, and we'd like to thank KCCR for giving us the opportunity to do our groundbreaking research. Thank you very much.
Thank you so much, Prof. Akim and Prof. Alex, Deborah, and compliments to Dr. Linda Deborah and Professor Ohinie J. On to the final one. There's a very popular saying by Shakespeare, to be or not to be. Now this one is TB or not TB. What 15 years of collaboration with KCCR taught us about immunopathology in mycobacterial diseases. Please help me welcome Professor Mark Jacobson. A round of applause. And I think I have to extend compliment to Professor Alice Osudabo, our very own Pro VC, and Professor Richard Phillips. All right, Prof.
Yeah, thank you very much for this kind introduction. Hope you can hear me. Hope that works. Okay, so yeah, it's a pleasure to be here, and I really feel honored to present some of the collaborations that we have here together with the KCCR. So I'm from the University Hospital in Düsseldorf, actually the Children's Hospital, and we work now for about, so me personally, for about 15 years in collaboration with different partners at the KCCR. And these were, is now Richard Phillips and formerly Alice Owusu-Dabu. And yeah, actually, I think it's a great meeting today here. I love to see so many former and current colleagues and also friends. So I think that's really great to meet here. So, and I would give you a short history how everything started for me, and actually the collaboration with African partners did not start when I was at the BNI, but before, when I was in Berlin at the Max Planck Institute for Infection Biology in the department of Stefan Kaufmann. And there was this rich guy, you all know, that supported our research, and he funded studies in Africa about TB, HIV co-infection and the influence of on the immune response. And yeah, there were many African partners, as you can see here, it's very small, and also partners from US and from Europe, but unfortunately, actually Ghana was not involved. But for me, it was really an important experience because I got so much insight into African country needs, African research, and quite different experience that we have in Europe, of course. Yeah, but then I changed actually to the Bernhard Nocht Institute for Tropical Medicine, and actually to the department of Bernhard Fleischer. As you can see here, I got immediately involved also in a mycobacterial disease as tuberculosis. I come to this, it's also a mycobacterial disease, of course, but this was about Buruli ulcer, which is of course a disease especially affecting individuals here in Ghana. And as you can see here, these two projects funded by the European Union, they were central also for the identification of immune biomarkers in Buruli ulcer disease. And Richard, you see already yourself, you recognize yourself, I think it's very small. And also you can see my former boss and mentor, Bernhard Fleischer, on this slide. So that was when everything started. But actually, I also wanted to go on with my research on tuberculosis, and therefore I went to Bernhard Fleischer because it was quite difficult to do this in Germany. And actually I remembered very clearly, he said to me, okay, Alice is here in Hamburg, you have to meet Alice. And actually I did that, and Alice, who is Alice Owusu-Dabu, as you can see here, was quite interested in doing research together with me, and that was really the initiation of our tuberculosis project here in Ghana. And the other one you may have also seen today here is Anthony Efuma-J-Avua, who was actually the first PhD student in this collaboration project. And actually another one, very small here, that is actually the funder from the DAHW Foundation in Germany who funded these projects. Yeah, in 2011 actually I went to Düsseldorf, and for me it was obviously the question, is this the end of this collaboration? Because obviously it's strongly connected to the BNI in Hamburg, but it was quite fortunate because at that time Richard Phillips had some interesting finding, and this interesting finding was that he found Manzanella persistens in serum samples of Buruli ulcer patients, and that was quite interesting. We also saw some association, and we've asked ourselves, okay, could nematodes influence disease course, immune response, or something like that? And then we started a really great collaboration with the group of Achim Hörauf, and you can see here our, I think it was a meeting in Tanzania in 2014. You can see here Achim Hörauf, our Cameroon partner, Samuel Wanji, Laura, Ken, you may know, and Richard, of course, and me. And yeah, that was really the start of a, as Achim already told us, it's difficult to get funding for these projects, and we got one project after another, starting with MAP-TB and then, with MAP2-CO, Manzanella and 2 co-infections, and then Manzanella in tuberculosis patients. Yeah, this is of course our team that is also so larger team. I will come to that, in this case, Linda Batzadipra and Dorcas Owusu. Okay, so but yeah, what we really did during the last 10 years as part of this project, we've strengthened our infrastructure here. And as also mentioned by others, the aim is really to finally transfer all the methods, all assays that we are doing to perform this directly at KCCR, and not as it started most of the analyses at the Bernhard Nocht Institute or now in Düsseldorf. And for this we established really a lot of and contributed to infrastructure here. This was about patient recruitment. As you can see here, we can transport our samples from distant places. We have a great team now at the KCCR. We have a great immunology lab, also with multi-color flow cytometer here, BSL-3 labs, and we are also established a lot of diagnostic capacity here at the KCCR. That was everything, of course, not possible without the help of especially Richard Phillips here in the Institute. So that was a bit about the background. Now let me give you just a short introduction to tuberculosis. I'm sure you are all aware about this disease, and tuberculosis is still, so caused by Mycobacterium tuberculosis, obviously not a tropical disease, but this disease nevertheless affects many individuals. Right now, the numbers from 2021 is that more than 10 million TB cases occurred in 2021, with more than 1.4 million deaths, only the HIV negative. But what I wanted to say about tropical diseases, of course, TB is not a tropical disease, but if you look for the landscape of tuberculosis, and here this is the changed landscape of the world, just about the numbers of tuberculosis patients, and as you can see, the western part of Europe, North America almost completely disappeared. Why? Especially Africa, Sub-Saharan Africa, and also India, other Asian countries become very large. So it is not a tropical disease, of course, but there are really the burden is much higher in this country and in others. And as you can see here also from the new report, those numbers go down in Europe during the past year, while they are still increasing in the African region, including Ghana. Yeah, I'm an, would say, hardcore immunologist, but I don't go into details too much of immunology because I don't think there are too many hardcore immunologists around, but one needs to know how a disease develops. And of course, not everybody who is in contact to a tuberculosis patient who gets infected by aerosol is developing a tuberculosis. If somebody gets detected, you can detect the immune response against Mycobacterium tuberculosis within about 6 weeks. And this immune response, that's what we know, largely protects against disease progression in the vast majority of individuals. And we know very much, or we know a lot about the granuloma structure in the lung that forms, and also about the immune structures and immune cells which are involved in protection against tuberculosis disease progression. but just in few cases. So we are talking about 10% in adults within the first 2 years. In children, it's much higher, with up to 40% in very young infants. You see so loss of immune control, some kind of immune disturbance that you can see here. You can hardly see here that there's of course also strong proliferation of the pathogen and dissemination of the pathogen in these patients. So the question that we want to answer is what causes this kind of immune disturbance and finally leads to disease progression in these tuberculosis patients. Yeah, and then I selected a study. I mean, there are so many studies out about tuberculosis and immune biomarkers, but I liked the study because they really wrap up many studies from all around the world, as you can see here, different African countries, European and other South American countries, and they did really a lot of research on immune biomarkers, multiplex, sequencing, and so on. And what I believe came out of this study? Only one parameter they could identify that was common and seen in all of these studies that was somehow associated with the risk to develop tuberculosis. My question to you, do you believe that we should all only look for NK cells now? If we don't believe that we should look for NK cells, we may think about why can it be the case that you only identify one parameter, although there are so many factors influencing. So we are quite sure there are many factors that are influencing susceptibility, and all the studies, animal models, genetic studies clearly show that there are multiple parameters like interferon gamma, like Th1, so T helper type 1 cells, and so on and so forth. And we think that it's important to look for all these different populations. And here it comes into place that we did a lot of studies in one population here around Kumasi. And we always, and I come to that, do different assays on these immune cells to see how different factors which may influence disease come together to finally may lead to immune pathology. And for sure, as I said, T cells are important, monocytes, the host cells, or macrophages, the host cells of M. tuberculosis are important, but what we identified during the last 2 or 3 years, that also obviously the cytokine milieu, what we call cytokine milieu is just what we measure in the plasma, and obviously the plasma of humans is a good place to look for biomarkers of this disease. It's of course lung disease, but obviously has some disseminated changes in immune pathology. And also, of course, other immune cells play a role. But there are also other factors. There are not only the different immune cell populations that contribute. One important factor, of course, are environmental factors like co-infections. Immune genetic factors play an important role, and we heard about that today. Behavior and social factors like nutrition play an important role, and also other comorbidities, maybe also other inflammatory autoimmune disease may play a role in the pathology. So we did some studies just shortly mentioned, and I said this nice study together with the group of Achim Hörauf looking for the influence of Manzonella perstans, and just the only point I would like to make why the co-infection itself was not strongly, at least in our cohort, affecting immune response. When we treated these individuals which were infected with Manzonella perstans and co-infected with mycobacteria, we treated them with doxycycline together with Achim Hörauf, then we saw very strong and sustainable changes in the immune response in these patients. So this may indicate maybe also other secondary effects of antibiotics, but of course removal changes the immune response may change also immune pathology in other diseases. Immune genetic factors, I was not aware that I would hear that much about immune genetics because we went completely the other way around. During our immune assays, we identified that the IL-7, the interleukin-7 receptor, was strongly affected on the immune cells in the serum. So there's a soluble IL-7 receptor, and therefore we also look for immune cells. And what was interesting, for the genetics, What was interesting about the IL-7 receptor is that we could identify two polymorphisms. These two polymorphisms were are only prevalent in the African population and in no other population around the world. But 20% of the African population here in Ghana carries this mutation, which was associated with alternative splicing of the IL-7 receptor, which is important for T-cell functions, and as we now know, also for monocyte functions. So I think, although if we look for all the genes and for global studies, these are not maybe, we don't see any association. That does not mean that looking for individual candidate genes and polymorphisms, that they would not affect immune pathology. Yeah, I very briefly go through our approach, but I think it's really important to understand how we try to develop our stories. So I love descriptive studies. Usually, if people talk about descriptive studies, they say, okay, it's the low level of science, you need more than this. But I think really detailed descriptive studies, what happens during immune pathology in patients can really be helpful, especially if you not look only for one population, but if you look for, or I come to that, but if you look for multiple parameters. So we did case control studies, did interventional studies, look for the treatment efficacy, and look for disease progression in infected ones to identify relevant parameters. And as said, looking for multiple immune cell and serum parameters. That's what we did. We combined this always with functional analyses, so like monocyte microbacterial kill assay or tuberculosis-specific T-cell assay that we perform. And we do a new, and I come to this briefly, a serum milieu assay to see what the effect of serum milieu on the immune response is. And in this case, also look for cytokine receptor signaling to look for important candidates. And then we have certain immune modulation assays available, lentiviral transduction and monocyte cell line assays to further go into detail of the function. And I showed this for two reasons. The first reason is we combine all this, we do this concomitantly in the same study to really not only look for changes, but at the same time try to identify really the pathways and the interaction between these different functional and differentially regulated markers. The other reason why I show you, so when we started, only the descriptive studies are done here at the KCCR, but our aim is now, in a new project that is now studied, to do also the functional and the immune modulation assays here at the KCCR to really strengthen capacity of the KCCR. So that will help, hopefully, also that the staff and the future scientists will see their future here at KCCR, and that they can really build on their science also for the future. Yeah, just one example that you don't believe that I'm just talking about nothing what is substantial. We did, as I said, immune phenotyping, and we started with T cells, but finally we ended up with monocytes in the peripheral blood. I was thinking, okay, monocytes usually would not have contact to TB, at least not in the peripheral blood. They migrate into the lung, and so what should be different? But others describe differences, and that's what we also found. So you find different, if you have here, TB patients controls, and what it means is just so many parameters at the same time analyzed and just reduced to two parameters showing in one graph. And the density, and so each of these are dots, very small dots here, the density of the dots and the distance between two dots tells you something about the similarity of two cells, if they are similar or dissimilar. And you can see here already, here are the TB patients, here's a big cloud of cells, while there are no cells in the controls. That's how you interpret these data. And if you come to the markers, which are finally so inducing these differences, you can find these markers which are different. You do not need to care about them if you are not an immunologist. Maybe it's for some of you important that this is an inhibitory receptor, which may be important also for people working with siglets. But for us interesting was, can we somehow model these differences also if we use serum from these patients and the contacts. And that's what we did is we used reference monocytes just from a healthy individual which were enriched, and then co-incubate with plasma samples from TB patients and controls. So in this case about 30 samples in each study group. And if you co-incubate then the reference monocytes and look for their phenotype, as here for the direct phenotyping of the patients, then you can see here, you find strong similarities for some markers in the upregulation if these monocytes from healthy individuals are co-cultured with serum samples. I don't go into detail, this is just submitted for publications. What is really interesting is that that really complex signaling pathways were induced in these monocytes from the reference. So it somehow indicates it is not the one cytokine, it is not the IL-6, it's not the IL-10, it's not whatever IL-1 beta, but it's a combination of different cytokines. And we hope that we somehow have sensor with these monocytes to detect the changes in the TB plasma. And that might also be of value for other diseases like COVID-19 or other inflammatory diseases. Yeah. I don't go too much into detail. This is just to summarize the findings that we concluded from all these studies that have all been done here, all with Richard Phillips and Professor Alice Uluvuzu-Dabu, and we found many differences, which all are induced by different serum factors like IL-6, IL-10, but there must be more. And we saw also changes in the functionality of these T cells. You see here the IL-7 receptor, IL-7 strongly affected. And just give you an example why this could be of relevance. As you can see here, the changes, the impaired T cell response to mitogen that we see, which we could directly associate to to IL-6 overexpression in the serum. This is of relevance for negative immune tests for tuberculosis. So the quantiferon gets negative just because of these high IL-6 levels. Yeah. Just wanted to mention there are so many students. I don't think that we have the number that Achim Hörhoff and Alex Debra presented, but we are on our way. There are so many students from Ghana coming to Germany, starting with Anthony. There were medical students like Kerstin Harling and Christian Lundhoff who came here to the KCCR and did their studies here, at least in part. And there was a big story of success. Ernest Adankwa, who is here in the audience, who performed his PhD in Düsseldorf in our graduate school for infectious diseases and is now back here in Ghana as a lecturer and also heading the administration of the lab. So really something, yeah, for us a story of success, and I think for him too. And we have new PhD students, Hubert Senanua Hall from Ghana, from Kumasi, and another one to come in the near future. Just short to mention, because I'm not talking about other projects, but we are extending our view here at the KCCR, now also looking for non-communicable diseases. One very interesting, of course, disease, type 1 diabetes, and this is the project by Julia Seifert, who is also here in the audience. Just summarizing, I would not talk about that. Please visit her poster, the poster of the Stop T1D project funded by the Eminöta Foundation, and then you will learn more about differences in the manifestation of this disease between Germany and Africa, which might be of relevance. Yeah, and we have a new project to start, which is, we call it the NOTME. NOTME stands for non-tuberculosis mycobacterial infection, which happens in more than 10% of all TB patients here in Ghana and throughout Africa. This is the case, so presumptive tuberculosis patients, which turn out to be atypical tuberculosis, atypical mycobacteria infected, and we have a special mycobacterium fortuitum identified here in Ghana, which made up the largest proportion of non-tuberculosis infected individuals. And the aim is to look, first of all, also to improve the diagnostics for this atypical mycobacteria, and to look for immune parameters which tell us something about susceptibility if these patients are highly susceptibility. Yeah, like actually for Bonn, and we followed them also wanted to get a bit more of institutional support of our collaboration, and so now on its way a memorandum of agreement between the KNUST and the University in Düsseldorf, and I am strongly working that this is also some practical or has some practical impact. Yeah, as said, nothing would have been possible without our collaborators. This is our group, actually a photo from 2021 from the MAPTB group, and I almost mentioned the majority of them. Yeah, nothing would have been possible without their help and without their support. And this is actually my smaller group, if I would say it that way, in Düsseldorf. But you can see here Wilfried Anyagiai, Hubert Senanua-Hor, both working. So one working on the Stop T1D project, one on is again graduate school PhD student, and I have actually another Rwandan student, nothing to do with the KCCR. But yeah, with this, I thank you for your attention.
Well, thank you so much, Professor Jacobsen. All right, I think it's been a long day today, and I would like all of you to give yourselves a round of applause. It's been a very successful first day. I'm going to call on the chairman for this afternoon's session, Professor Christian Ajari, to give his closing remarks. A round of applause for him.
Thank you very much. As Anita said, you spend a lot of time here. Some of us, or majority of us, we are tired. So the conventional way of summarizing the presentation may not happen. I will just give my brief closing remarks. But just for the sake of those who want to ask questions, just 3 questions or comments. Together, 3, in case we have anybody who would like to ask a question. Otherwise, I will go ahead with my brief closing remarks. Thank you very much for your cooperation. So at least you have been given the opportunity to ask questions, not the usual way. We've had 5 presentations that have indicated that the collaboration between the German government and that of our government that started in 1993 has achieved a lot. First, I will start with the fact that most of the areas or the research areas or the disease conditions that have been investigated so far affect those of us in the tropical region or Africa. So the research outcomes are helping us to change policy and to help manage the various disease conditions. Secondly, out of this collaboration, we've been able to train a lot of our people, and then through collaboration, we've been able to also get a lot of knowledge and skills out of this engagement. Finally, with this collaboration, we've been able to publish a lot. And through this publication, some of our staff have been promoted, others have gotten the appointment within this university and other universities, even outside the country. We are hoping that moving forward, the next 25 years, maybe some of us may not be around, but I'm hoping majority will be around. We are going to celebrate the center with a lot of achievement, more than what we've done so far. With this, I clap for you, the fantastic audience, and then the presenters. And I wish you well in the next few days of the celebration. Thank you.
Thank you so much for a very well-chaired session. A quick announcement. Tomorrow we'll continue with the scientific symposium. It will start right here at 9 a.m. at the Great Hall. We'll be here at 9 a.m. So please be here early with the theme, Improving the Quality and Quantity of Scientific Research in Africa. There'll also be some tour at the KCCR laboratories in the afternoon, and later in the evening, there will be the party and live band. So tomorrow promises to be really interesting. Don't miss any part of it. We'd like to acknowledge your patience, your presence, and your time for today. It's very much appreciated. A round of applause for all of you. Thank you so much. We wish you a really restful evening. Sleep well, sleep soundly, enjoy our beautiful campus in the process. We'll see you in the morning. Thank you very much. Thank you very much. Good night.